Okay, so today is our 10th lecture. So it's going to be today's and this Wednesday is going to be a combination of lectures on transcriptomics. So they're going to be tightly linked. So today we're going to talk about the platform first, like how, which technology that we can use to study gene expression. We will talk about some statistical model for gene expression data, but that will be the focus for our next lecture. We'll focus on the differential expression analysis. Okay, so today we're going to mention three main techniques: oligonucleotide microarray, or shortly microarray, Nanostring, and RNA sequencing. Okay. So, like the first question that we might ask is why transcriptomics is so popular compared to other omics. And that's because it's easy to quantify in the sense that if you compare transcriptomics to proteomics, as we will cover in a few lectures, proteomics, the study of protein, is not that easy. We can no longer do polymerization reaction. We can no longer amplify the protein. So there is a lot of both technical and analysis difficulties. So studying gene expression is much simpler than protein expression. And compared to genomics and epigenomics that we already covered, if we want to understand the effects of mutation or change in epigenetics on the cells, we need to understand how it affects the gene expression, the activity of the gene. So transcriptomics is like a midway between closer to function and phenotype than genomics and epigenetics, but not as close as protein, but it's easier to study than protein. So it's at a point where it's a perfect trade-off between the difficulty and how much we can interpret function and phenotype. Okay. So the first technology that we are going to look at is really old, but still kind of in use today because it's cheap, and that's the microarray technology. So what we imagine as a microarray is you might have a chip, like a surface, and on that surface, people design a certain DNA sequence or RNA sequence, and they attach it on the chip. So each location, each kind of pixel or each cells on the chip correspond to one design or one target nucleotide. So that's why we call this oligonucleotide microarray. It consists of oligonucleotide, so like an oligomer of nucleotide, and it's on an array, and it's very small. So that's why it's named this way. So just for your curiosity, there are many ways that we can make or manufacture a microarray to have the amino acid sequence, the nucleotide sequence that we want. For example, we might try to synthesize the oligonucleotide on each cells, right, using reaction, and we control the reaction so that we add A, T, C, or G on specific cells by kind of controlling the reaction maybe with a filter, so that when we shine the light to activate the polymerization reaction, if we have a filter that says, I only want A to be incorporated in certain cells, like we can filter out the other cells. So if we create a mask like this, then we can control the polymerization reaction on specific location, and we can construct a specific oligonucleotide, oligomer that way. Another way, we can also kind of inject specific nucleotide directly onto each cells. So this is more modern technique. Okay, so just to give you like an overview of how we can construct such thing, but usually that's not our task. Usually we can buy pre-made microarray and use it for our study. But if you ever have to study like a non-model organisms and you have your own nucleotide sequence that you want to target, you can ask company to create like a custom microarray for you. So we want to talk a little bit about the design for the array because it will comes into play when we analyze the data. So first, the microarray, it's not that simple. It's not like we have all the sequence we want to target and then we just put it on the array. because there can be other problems. For example, if your oligonucleotide is not specific, like this also happens when you design primer for your experiment. If your primer is not specific, you're going to amplify more than just the gene of interest, you're going to amplify other DNA as well. So the same for nucleotide microarray. If your nucleotide is not specific to a gene, it will also bind to other genes, maybe other fragments from the genomes. So that can create a false positive signal. And sometimes your oligonucleotide can bind among themselves, like if they have complementary DNA or RNA sequence. So you can have an unwanted interaction among your sequence. And sometimes the nucleotide itself can fold because nucleotide on the long strand can form secondary structure. So it can fold into itself, and this will no longer be active. It will not bind to your gene. So without good design, your array can have a lot of wasteful spot that doesn't detect anything, or it detects too many things, and it cannot give you a specific information. So just to summarize, so when people design a microarray, similar to how you design primer, you want it to be sensitive. So you want to know the target of the gene or the genomic region that you are going to study. Since this is for transcriptomics, we're going to analyze all the exon, the coding sequence. So we need to know the exon first, so that we can design probe nucleotide that's complementary to the exon. And just to make sure, like, because exon is long, we can also design probe to target different regions on the exon, so that we are sure that at least one of our probes will bind to it, so that we get the quantification for the gene. So that's to increase the sensitivity. We want to be able to detect genes that we want. But we also have to worry about specificity. We don't want the probe that we design, like primer, to be able to cross-react or cross-hybridize with other genes that we don't want. And these people can do quickly with BLAST. We know that we can search with a certain nucleotide sequence, like we can BLAST against the whole genome to see if it will match to multiple regions on the genome. And later on, we'll see some slides that talks about we not only want a nucleotide that can perfectly match to the exon, we want to also have some control, some negative control, that we introduce some mismatch, like we intentionally make the probe not perfectly complementary to the exon. And that will allow us to measure the non-specific binding. Because if we have only perfect probes, we have no idea if it will react with other sequence. But if we have slightly imperfect probes, we can compare the binding activity between the two, and we can determine how much is non-specific, how much is specific. And the rest would be technical issue that I already mentioned. We want to prevent secondary structure formation on our nucleotide. We don't want it to form a hairpin because this will not react with the gene, the exon. We also want to control the hybridization energy to be roughly the same throughout the array. This is similar to how you design primers. Like you want your primers to operate at the same temperature so that you can run the reaction and you get equal binding and equal hybridization throughout. And on the array, because we are going to have plenty of location to put our nucleotide. So people are going to put the same nucleotide several locations so that we can be sure that if our RNA sample, the concentration might differ across the chip. If we have probes only in one corner, we might get a bias where we might have low DNA concentration or high RNA concentration in certain locations. So in the design microarray, they're going to incorporate some redundancy. So we're going to put maybe the same probe at multiple locations on the chip. And then we can average the signal so that we get a better, more confident measurement. So we can put the redundant probes on the array. Okay. Just to show you that the position on the array can introduce biases in the binding in the concentration of RNA. So people found that on one microarray, even if you put in the same probe, you can have different intensity, different kind of hybridization, just because your sample is not your RNA molecules are not evenly distributed, and maybe the imaging techniques is not evenly distributed, so you don't get the same measurement throughout the array. So we need to do some correction for this. But luckily for you, most of these are already part of a standard bioinformatics tool, so you don't have to study the pattern and then model it yourself. But we can talk a little bit about the principle behind how we can correct for such kind of position-specific bias in the array data. Okay. What I want to show you next, so these are very old, you can tell from the font. So these are studies from back when people start to develop microarray. They have a lot of questions that they need to optimize. How long should the oligomer be? Do we want the probe to be 25 nucleotide, 50, 70, 100? We can kind of guess that if we have short nucleotide, it's too short, it's not going to be specific. But if it's too long, then multiple exons might be able to hybridize to that probe. So back then people have to kind of find the trade-off in between. And I think what they found is around 100 to 200 nucleotide. If you design probe of this length, you get a good enough sensitivity and specificity to your data. And as you have more concentration of your RNA, you want your intensity, like the hybridization, to scale with the concentration, so that you can get an accurate measurement of gene expression. But one limitation for microarray is that if you have very high concentration of your sample, at a certain point it's going to saturate. This is normal for any chemical reaction. Up to a certain point, the signal is not going to get stronger. This is where you get the same signal regardless of how much more RNA molecules you put in. So this is one limitation of microarray. You are not going to get a quantitative measurement for certain range of gene expression. But otherwise you're going to get an okay approximation of the gene expression. So this I already mentioned that when we want to study a gene, the gene is long, the exon is long. So there is no reason why we cannot design multiple probes for each gene, for each exon, so that we get more sensitivity. So back then people also tried, how many probes should we design for each gene? If you put in too many probes, we are going to lose out on the space on the microarray. We will be able to target less gene if we use 10, 20 probes per gene. But if we use only one or two, the data that we get might not be reliable, because it can be random noise or bias that can skew the measurement. So people try to find a balance. I think the balance is about 5 to 10, if I remember correctly, 5 to 10 probes per gene. And then you can average the signal from multiple probes to get a more stable estimate of the gene expression. Okay. And I mentioned that we not only design probes that are perfectly matched or perfectly complementary to the target RNA, we also people also try putting in some mismatch intentionally, so that when we have measurement from these two probes, we can compare the expression level that we get from the perfect match and the mismatch probe to estimate how much could be from non-specific interaction. Because for mismatch, we intentionally design that it's gonna bind to the RNA, but not that well. So the difference in signal can give us an idea of how much specific binding and non-specific binding there are. I'm not going into the specific formula for it, but just to give you an idea that when we have a lot of space on the microarray, we can design probes for different tasks. So probe for measuring the expression, and probes to control and adjust the measurement. So today, the microarray technology has improved a lot. now we don't have to run one sample per array. So back then you have to run each sample on a separate microarray. And one thing that you can imagine is you can have a technical bias. Different array might give you different level of signal just because of the fabrication or maybe the RNA concentration and other technical factors. But nowadays, we can run multiple samples. I'm not sure if it's up to 4 or 8 now, but you can run multiple samples together at the same time on the same microarray. So the idea is for each sample, the RNA molecules are going to be attached to different fluorescence. So let's say sample 1 attached to a green fluorescence tag, sample 2 attached to a red fluorescence tag. So when they hybridize to the microarray, because the same gene from these two samples, if they are the same gene, they have the same sequence. So they're going to bind to the same microarray spots. But because they are from different samples and they are tagged with different fluorescence, so when we activate the fluorescent signals at one location, certain location on the array, the amount of green signal and the amount of red signal that we get is going to be proportional, roughly, to the amount of RNA for that gene, the same position. From sample 1, if it's high in sample 1, then you get a bright green signal. If it's high in sample 2, you get a bright red signal. So when you overlap the two, you're going to get a gradient between red, yellow, orange, and green. So if you see a green signal, it means that your genes are highly expressed in sample 1 because the green signal kind of dominate the red colors. If you have yellow or orange, that means you have a roughly the same expression level, like blue and red mixed to create yellow and orange. But if you see bright red signal, this means that that gene is highly expressed in sample 2, but not in 1, because you have red signal dominate the green signal. And because you are running a single microarray, right, this minimizes the technical variance. We're going to see the same concept in proteomics, where when we have multiple protein samples, in the past, people have to process each protein sample through the mass spectrometry one at a time. And we have a lot of technical variance. But nowadays, there are techniques that we can combine, let's say, blood sample from 8 patients or 10 patients and put them together in one mass spectrometry runs. And we can deconvolute the signal afterward, like how we deconvolute the color to interpret whether it's high in 1 or high in 2 or equal. We can do the same with mass spectrometry data. Okay. So just for your information, microarray technology is not only applicable for transcriptomics. People have adapted this idea that we can put oligonucleotide on an array for other study. So let's look at that. Okay. So one of them is called comparative genome hybridization. So as the name suggests, we want to compare two genomes, maybe patient with certain disease and a healthy control. We want to know if in the patient with that disease, do they have a certain deletion of the genome or copy number variation of the genome. Like some region might be duplicated, some region might be lost. We want to compare between our patient and the control. How can we do that with microarray? So instead of designing the microarray, the oligonucleotide, instead of designing it for exon, like which we use to monitor gene expression, if we design it against every region of the genome, we design on every chromosome, against every chromosome. And then we tag DNA from patient with red fluorescent and DNA from control healthy person with green fluorescence, right? And we mix them together, run the array. If we see a spot with green, so this means that we have more DNA from the control than the patient. Having more DNA in the control means that in the patient must have lost that section of the DNA. So if there is a deletion in the patient, there will be less DNA from that part of the chromosome compared to the control. So the signal on the microarray will bias toward the green, which is the control. But if we see red color from the microarray, in this case, it means we have more DNA from that chromosome position in the patient than in the control. So So there must be a duplication or some gain in gene copy numbers in the patient. So for human genome or mouse genome or model organisms, where we understand the whole genome sequence, we can design nucleotides that target different parts of the chromosome. So we can use microarray techniques to analyze if the patient has a gain or loss of certain segment of the DNA without doing sequencing. So the idea that people does this is, like, you might think that if we do exome sequencing or whole genome sequencing, we also got this information. We can compare the read count to see if in the patient do we have more read count in certain location or low read count in certain location. But you have to understand that sequencing is expensive, especially if you want to sequence the whole genome. It's going to be more expensive than just exome sequencing. But with microarray, one good thing is, as let's say you pick human and you decide the nucleotide that target the entire human genome, you create this array. Everyone in the world can use. You don't have to run the sequencing again. You just produce this chip, this microarray, and then everyone can run their sample to get it. Okay. So that's one idea, to make microarray technology, which is cheaper than sequencing for tasks other than transcriptomics. People also try to use microarray for SNP genotyping, so like exome sequencing, for example. So the idea is we're going to use the nucleotide, the oligonucleotide on the array. You can think of it as a sequence, as a PCR primer. So if you have a gene that match to this nucleotide, like oligonucleotide, it's going to hybridize. And if you initiate a polymerization reaction, you will be able to sequence the rest of the gene. So basically they are doing a multiplex PCR amplification, but they are using this for genotyping by making sure that all the nucleotides that they design, like all the oligonucleotide sequences they design, are the sequence upstream of the SNP location. So their probes basically are going to recognize the DNA all the way right before the SNP location. So you can run the sequencing, the polymerization reaction, just one step, and you can sequence the SNPs. So they will design like different primers to target the upstream region of different SNPs and run the polymerization reaction and look at the signal for each location on the array. Right? So each location on the array will be targeted different SNPs. So when we do polymerization reaction and we see incorporation of certain bases on the array, we will know like, which SNPs, which location of the genome produce that reaction. Right? So we can tell the genotype for that position. So you can also imagine of this like in Illumina or 454 sequencing platform, where you have different kind of location on the flow cell, different location on the machine that sequence different DNA. But when we do next generation sequencing within the machine, right, we have no idea which location, at each location on the flow cell, which region of the genomes are being sequenced, right, because it's all random. But for microarray, because we are designing oligonucleotide specifically on each cell, we know exactly for each signal which region of the genome or which SNPs does it belong to. So people have adapted this for SNP genotyping. And similarly, like, you can couple this with other molecular assays, like bisulfite conversion. So if you design your probe all the way up to the CG island, and you do a bisulfite conversion that detect between modified or unmodified cytosine, you can do a polymerization reaction and see if you can incorporate a G or an A onto that location. And then you can compare between with the bisulfite and without the bisulfite. So you can identify DNA methylation. So these are like the same thing that you learn from sequencing lectures, but adapted to be made on microarray. And just to emphasize, like, the idea is that microarray and DNA sequencing actually are interchangeable in many situations. We can do genome sequencing, we can do SNP genotyping, we can do transcriptomics, we can do with either microarray or DNA sequencing. The difference is microarray, you decide it once, you decide the nucleotide, and then everyone can reuse. So it's cheap. Instead of everyone have to do sequencing again and again, someone spend the effort to decide the array, and you can sell it to other people. But the limitation is, because microarray by design, you have to fix all the nucleotide sequence. So you cannot detect anything that's unknown. You cannot detect anything new. And everything that you can detect are the things that you already targeted, for. So that's the trade-off. With sequencing, you can discover many new molecules, many new RNA, non-coding RNA, but it's expensive. But if your task, the things that you want to do, the experiment that you want to do, only involve known genes or maybe known region on the genome, if there is a microarray for that, that can reduce the cost a lot. Okay. So now that we know about the microarray technology, so let's see how we can handle or how people handle the data. So first, when you look at microarray data, they're going to give you detailed information for each position on the array, like X and Y coordinates. What's the probe? What's the nucleotide that's being put on the array? And some nucleotide might correspond to genes. So these are gene accession ID. But some nucleotide might be control. So they will tell you on each position of the array. Is it spot like for a certain gene, or is it for controlling the signal? So to give you a recap, so many sets are just simple statistics. So when we get a microarray data, you will see these probes, which are sometimes it doesn't make any sense. It's just a number. Sometimes they already gave you the genes that the probe mapped it to, so you can interpret that. But in some cases you don't. In some cases, you get the data that only have the probe ID. So you have to go to website and then try to identify like which nucleotide sequence were assigned on that probe. And you have to use BLAST to annotate the gene to the probe. But the good thing is, if you are using like a commercially available microarray, this mapping has already been done. So usually you get a table like this that map the probe ID to the gene ID, so that you can interpret it right away. Okay. And I already mentioned that the intensity, like the fluorescent signal that we are getting from the microarray, is not going to be homogeneous. So in the bioinformatics software, they're going to have to do some correction that depends on the position on the array, or depends on whether a perfect match probe or a mismatch probe. So in this lecture, we're going to talk a little bit about how we can correct for position-specific bias as an example of how we can formulate this problem. And one thing that I will emphasize is for intensity signal, like fluorescent signal or light signals, like these kind of intensity, people have found that they are going to follow a distribution called log normal distribution. Basically, if you look at the intensity data of any experiment, what it will look like is that you have going to have a lot of low intensity result, and then some pixels or some image with very high intensity. So the distribution is going to be skewed like this most of the time when you look at fluorescent signal or light signal. So people found that this kind of distribution, when you take the logarithm of the result of the signal, you usually end up with something that looks more or less normally distributed. So people name this log normal, which means that the distribution itself is not normal, but when you take a log transform of the data, you get a normal distribution. So these correspond to kind of intensity data. And also we will see for the mass spectrometry, when we count the number of ions for the protein, they are also going to follow the log normal distribution. And people in the past have tried to figure this out by fitting the actual signal that we get from the microarray. And they found that it can fit to the log normal distribution very well. So we're going to use this as our assumption, that the signal that we are getting is going to be log normally distributed. Okay. So what can we do with this kind of statistical knowledge? Let's try to use this to correct for position-specific noises. So I think we are now kind of familiar with this idea now that if we want to test, like if we want to remove certain effects, like certain noise, like we're going to make some hypothesis, and we're going to reproduce the noise by trying to reject the hypothesis whether the noise is distributed a certain way. So in this case, what we are going to use as a null hypothesis is that the noise that we are seeing is not depending on the position. So first, if we want to do a position-specific correction, first we want to prove that the noise are position-specific before we can do that. So the null hypothesis will be the noise in this array are all normal, or normally distributed after the log transform, or normally distributed, and it doesn't depend on where it is on the array. It's the same. We are just seeing some variation by chance. And our alternative hypothesis in this case is that the noise depends on the position. And to make it simple, we're going to make the position model here very simple. So the noise is going to scale with the x and y coordinate. So if you compare the two hypotheses, the variance of the noise we assume to be the same, but we're going to assume that the average noise level for the null hypothesis is mu, like some number, which is the same for any x and y. But for the alternative, we're going to assume that it's not just mu, it's not just a constant. It's going to have some factor times x and then plus some factor times y. In fact, it's not that simple, but we're going to use this to make the calculation simple. So we have two hypotheses. We have to compare, like which one is more believable. We have to reject one and favor the other. So let's start with the simpler one first, the null hypothesis. If we assume that all the noise that we are seeing are the same, have the same average kind of throughout, we can fit the data. So let's say on our probes, on our array, there are certain probes that are negative control. So these probes shouldn't bind to any DNA, any RNA. But yet we're still seeing some signal. So these are signals that we got from the array. Let's say we already got transformed. Because these are negative control, we assume that all of these are going to be noise, pure noise, like no signal at all, pure noise. And by our null hypothesis, we assume that it's normal distribution with this mean of mu and the variance of sigma squared. You don't have to worry about the symbol. Right? Just think of it as it's normally distributed. It's going to have some mean and some variance. So we know from statistics review that if we apply the maximum likelihood technique, we have some data. We have some parameters that we don't know yet. We don't know yet what mu is, what sigma square is. But we can calculate the likelihood. Let's say if you look up the probability density function for normal distribution, but if you forgot, it's okay. We just want to appreciate the fact that if we have the data and we have some assumption of the distribution, in this case normal distribution, we can write the likelihood, probability of observing this data given the parameters. And in this case, parameter is the normal distribution. And we will have some expression, some mathematical expression that looks maybe scary. But by the maximum likelihood principle, we want to identify mu and sigma square that make this term the largest. We want to maximize this term. So we can take the derivative of this with respect to mu and sigma, and then set it to 0, and then so forth, which we are not going to do. But we want you to appreciate the fact that this can be done. And with not much difficulty, we can do it. So we know that under the null hypothesis, we can estimate mu and sigma from the data. But for the alternative hypothesis, it's going to be more interesting because now the average signal, the average noise level is now going to have both the x and y coordinates in the formula, not just like a number. But again, we can apply the concept of maximizing certain likelihood. But in this case, I'm going to write it in a different way so that we see a different formulation. So let's say we have like the noises, like all the signal here, like the intensity in subscript I here. These are intensity of the negative probes. So these are all noise. But because negative probes are located in different locations on the array, so each of them will have its own position at xi and yi, like different position on the image. So we have the data for each negative probe. We know the location, and we have the alternative hypothesis that the average should depend on the position like this, as a linear function. So we want to estimate A, B, and mu. These are three things that we don't know. X and Y are just position. Right? We can look up on the from the metadata of the array. We don't know A, B, and mu, the blue parameters. So what we want to try is, can we try to identify A, B, and mu by trying to minimize this term? So what this term? We have the intensity at each location. We have this formula which expresses our hypothesis that if a negative probe is at this position xi and yi, this would be the noise that we expect, axi plus byi plus mu. This is the noise that we expect from this probe. We have some expectation, but ni is our observation. So we want to make sure that our expectation and our observation match as much as possible. So we subtract them, and we take the square, and we sum them. So if this term is small, that means the data and our expectation are similar. If this term is large, this means that our observation and our expectation doesn't match. So we want to minimize this term so that our choice of A, B, and mu makes it so that our hypothesis of linear effect match to the observation as much as possible. So again, we can do calculus. We can take this term, differentiate it with respect to these three parameters, these three unknowns. So each will give you something like this. And if you look at it closely, it's going to turn out it's not that scary. These are all linear, right? These are all linear functions of A, B, and mu. Because x and n and y, these are numbers. Once you have your observation from the microarray, you have the log intensity as numbers. You have the position of the probe as numbers. So the n, the x, and the y are just going to be numbers that you get. The only parameters that you have to solve are A, B, and mu. And now you have three linear equations, right, with three unknowns. Right? You know how to solve it. We are not going to do it here, but you know how to solve it. So basically, we can calculate on one hand, we can fit our data to the null hypothesis, and we get mu, we get sigma squared. From the alternative hypothesis that we have some position-specific effects on the noise, we can also estimate our A, B, and mu, and then we can get the sigma. So now we know that we have the estimate from the two hypotheses. Now we can compare them. Which one fits to the data better? So we have the likelihood under the null hypothesis, where we estimate these two parameters. And we have the likelihood for the alternative hypothesis, where we estimate 4 parameters. We have 2 more parameters here for the x and the y coordinate. So there are many ways that you can compare these two. I think in the past lecture, we mentioned something about likelihood ratio tests. So for this lecture, I'm going to add more techniques, more tools that you can use. Information criterion, you saw it in the assignment for the phylogenetic question. We want to identify nucleotide substitution model that fits the data better. And different nucleotide substitution model have different number of parameters. So in this case, we have two models that are trying to fit the noise of our microarray. And one of them has 2 parameters, the other one has 4. And we know that both have mu and both have sigma. So the alternative hypothesis is more complex. It has 2 more parameters. So with information criterion, so these are statistical idea that if you want to reject the simpler model of the null hypothesis, you want to be able to show that the more complex model, like the alternative hypothesis, does the likelihood increase a lot? Like that's the first criteria. We want the alternative hypothesis to have a much better likelihood, to fit the data much better. But we also don't want the complex hypothesis to be too complex. We don't want it to have 100 parameters. So these information criteria are going to balance between 2 things. And you will see K is the number of parameters, and likelihood, which is how well the hypothesis fit to the data. And they're going to compete with each other. So you're going to see the minus symbol. So both aggregate information criterion and Bayesian information criterion. This score is going to be high if you have a complex model, like your K is high, and you have bad likelihood. So high information criterion means you have a bad model. You have more parameters, but you have bad likelihood. But if this information criterion, if the score is low, this means you either have low number of parameters, or your model is simple, or you have a high likelihood, it's negative. You have a high likelihood, your model fit the data better. So you can use this calculation either by 2 times the number of parameters minus 2 times the log of the likelihood, or the log of the, n here is the number of data, number of negative probes, times the number of parameters subtracted by 2 times the log likelihood. So you want to prefer either the null hypothesis or the alternative hypothesis. Which one that gives the lower score? And you will pick that one. Okay. I think we talked about likelihood ratio test before, and I want to show that it's related to the information criterion. So when we perform nested model testing or likelihood ratio test that you have probably seen from statistics class, the score that you are going to use to calculate the p-value is going to be -2 times the difference in the log likelihood between the 2 models. And when you add this formula carefully, it looks like you are subtracting the information criterion from the 2 models. But you kind of just remove the number of parameters. You focus on the likelihood. So the delta log likelihood is like you calculate the information criterion for the 2 hypotheses and then subtract them. So you get the -2 delta log likelihood from the latter term. So what happened to the number of parameters? When you perform likelihood ratio test or nested model testing, you're going to consider this score with a chi-square distribution. And you're going to have to select your degree of freedom, the delta degree of freedom. And that's the difference in number of parameters. So basically, information criterion is the same, the idea is the same as likelihood ratio test, except that they combine the difference in degree of freedom and the difference in likelihood together as a single score. But when you perform likelihood ratio test, you calculate the difference in likelihood, and then you do chi-square test with the difference in number of parameters. So it's the same concept, but formulated in different way. And now you know, like all these things that people use differently, actually they have the same principle. Okay, so that's what I want to talk about the calculation that we perform to correct the noise in microarray. We're going to save the differential expression analysis for the next lecture so that it goes together with the RNA sequencing data and everything. So for today, I'm going to introduce the platform first. So after microarray, the other platform that some of you might have seen, but might be new to some of you, is called Nanostring. So this is a very new technology, but the concept is not that new. So basically, we want to have a very accurate measurement of gene expression. Like we mentioned that microarray is not that accurate because the hybridization and the fluorescent signal sometimes doesn't scale all the way, right? Sometimes it's saturated. And when we do sequencing, like we mentioned from before, that when we do sequencing, like we got the read count. The read count can be affected by PCR amplification. It can be affected by other bias. The best thing that you can do if you want to measure gene expression or expression of anything is to count the number of molecules. If you can look through the microscope and count individual RNA, that's the most accurate measurement. But you cannot look through the microscope and count the actual number of RNA. So what Nanostring people decide is that they're going to create, like, so for protein we have antibody pull down, right? So in this case, because it's nucleotide, right, we can target nucleotide with probes, complementary probes. But if we target the nucleotide with just complementary probes, then we cannot see, right? We cannot count. So in this case, the probe that they design have two regions. The first region is complementary to the gene that we are looking at. The second region is specific to a fluorescent reporter molecules. So that when we have a certain gene is captured by this probe, it's going to also emit this fluorescent pattern. And not just one color, they use 6 to 8 colors. So this means that when you look in your data, like this, could be very small, each spot is going to be a single RNA molecule. And which RNA, which gene, like which species of RNA, you're going to be able to identify based on the color coding of the fluorescence. So for example, if it's like red, blue, yellow, green, red, green, like this is FOX5. So this is already pre-decided by the company. So when you have captured this fluorescent image of your sample, you can count, use software to count how many molecules of FOX5, how many molecules of XLSA are present in your sample. Okay. So again, like, this is a trade-off between very accurate measurement, but if we have to have like a fluorescent like pattern for each gene, you're not going to be able to detect many genes because you are going to run out of fluorescent pattern. So this technology, the limitation, the trade-off is it can only target up to 800 targets, and this already exhausts all the possibility of nucleotide, all the possibility of fluorescent patterns that they can produce. And similar to microarray, you're going to be restricted to whichever genes that you design the probe for. So in this case, when you go to Nanostring company, they're going to ask if your research question already fall into some popular gene list that they have. Like if you're studying human cancer or mouse cancer, they already have the probes designed for cancer-related genes. And they have the probe designed for study in dogs, for example, and maybe for immunology. But if you have a new system, like a new cell type, a new tissue type, they can also work with you to develop the panel. I heard that this can be expensive. But again, once you develop this panel, like everyone can use. So it's the same idea as a microarray. So since this is different from sequencing in the sense that it's imaging, this is fluorescent imaging, similar to microarray, but it's more quantitative. So in nanopore system, there are many quality checks that I think is interesting to introduce to you. So first, they're going to analyze if your probes work as intended. So by working here means that do they bind to your RNA? So they're going to examine different regions. Like if you draw a square like on the image, they're going to look at different regions, and they're going to see if you can see fluorescent signal in those regions. If your regions are all black, that means your probe somehow doesn't bind to your RNA. or maybe your RNA, you have a very low RNA concentration. So that's the first quality check whether you have good signals or not. And they have some expectation that if your RNA concentration is what they want, what you want it to be, they are expecting about 1.1 to 2 molecules per square micron in that data. Because you don't want the fluorescence to be clouded. If you have multiple colors on top of each other, you cannot decode which gene they are. So for Nanostring analysis, they're going to have some expected RNA concentration that you should produce, and that would produce a certain density of fluorescence in the image. So they're going to check for that. Do we have the right amount of signal given the target RNA concentration that we put in? The other two steps is the how we can normalize the intensity, the numbers. Because even though this is like the most accurate measurement, like the number of molecules, we're going to have to accept that not all RNA is going to bind to the probe and show up here. Otherwise, we're going to have millions of fluorescent signals, and we cannot count them. We cannot decode them all. So in this case, only some part of the molecules will be bound, and the signal will be shown. So we are kind of subsampling. We are going to sample only part of the molecules. So there can be some randomness. If you have a low abundant gene, your signal might be high or low by chance, depending on how many probes happen to bind to your RNA if they are in low concentration. And they also have to worry about non-specific binding. Always, like when we design probes or primer, like we always worry about non-specific binding. So for microarray, they have a specific spot for the control. In the case of Nanostring, because everything is in solution, there is no array. This all happens in solution. You put in your probe and your RNA sample in the tube, and everything binds and then emit the fluorescence. So they're going to have to do something different. So for negative control, this is simple. If you design probe that shouldn't bind to any DNA, you design probe that doesn't match to anything in the human genome, those would be negative control. You shouldn't get any fluorescent patterns from those probes. For the positive control, so this is the opportunity that Nanostring can do. Because they are going to design their own probes, right? So they also design additional synthetic probes. So these are DNA sequences that do not appear in the genome, but instead of putting them and making them like a negative control, the Nanostring kit also have the complementary RNA for this probe with specific concentration from 0.125 femtomolar to all the way to 128 femtomolar. So they're going to spike this sample into your RNA sample. So these probes, these 6 probes, the intensity that the number of molecules that you are going to count should scale from this concentration to this concentration. So you have a ladder that you can normalize your data against. This is something that is difficult to do with other techniques because you cannot control the signal. In this case, you have the probe, right? So you can put in your spike ladder control. So let's see how they do it. So Nanostring create their own software called Encounter or Ensolver. But all the principles that we're going to talk about next, you can do it in Excel. And actually, in last year assignment, I asked the students to perform this in Excel. So let's see what they are. Let's apply our knowledge in statistics to solve this problem. So let's start with the negative control first. So we have, let's say, 8 negative control. So these are probes that shouldn't bind to anything. But yet we still see some molecules in the image. So these are non-specific signals. There are 2 ways that people think about these kind of completely noise distribution. We can think of it as a background signal. So if we have other probes and the signal from that probe are in the same range, like let's say we have a probe that should target a gene, but the signal is like 10 to 20, you can remove those. you can say that those probes, it shows signals that are lower than noise, so we shouldn't trust them. So in this case, we can threshold. We can say that any probe that have less than, let's say, 20 count, we are not going to analyze. We are not going to trust it. Another idea is, so if you think that every probe have the same non-specific signal, so basically you imagine that every signal are going to be plus 20. Every probe have a plus 20 signal. So in this case, you can also use a model called background subtraction. So we're going to subtract 20 from every probe. So that's also possible. So in this tool, they're going to offer you the choice because in real life, no one knows which one is best. So, but they offer you the choice that if you believe that, okay, I'm gonna discard all the probe with low signal, then we do thresholding. If we believe that everything has a non-specific binding plus specific binding, then we do subtraction, minus everything by 20. And in this case, they're going to estimate the amount of background noise based on the average of all these 8 controls. So that's, we're going to go back to that notion in a bit. So for the background noise, negative control, they're calculating the average, the arithmetic means of the signal. Let's look at the positive control. So we mentioned that they have 6 positive control probes, and these correspond to synthetic DNA that are spiked in, so they're put in by the kit, not from our RNA sample, they're part of the kit, and they have concentration that range from 0.125 all the way to 128. So they scale kind of twofold, maybe more than twofold, several times. And these are the data that we get from our sample. So let's say we have 24,000 count for the highest positive probes and only 50 for the lowest one. Because we know that these probes correspond to synthetic DNA that are spiked in with a specific concentration, so we have an expectation that this scale should follow the change in concentration. So what we can do is we can divide all these count by the expected concentration, like 128 femtomolar, maybe 64 femtomolar. So we get the ratio, multiple ratios. So these are deviation from the concentration that we observed. If we want to collect the data, we should use this ratio as a factor that we can multiply or divide to the data. So in this case, they're going to calculate the ratio between this count with the concentration for all the 6 positive control. And they're going to take what's called a geometric mean, not an arithmetic mean. They can take a geometric mean. And we'll come back to this, why we use arithmetic mean for the noise, but we use geometric mean for the ratio between actual count and the concentration. Okay, let's keep that thought in mind for a little bit. So another control that Nanostring use is the housekeeping gene. So in biology, we know that there are some genes that have very basic function that maintain the cells that are so essential that if you have any kind of disruption of this gene, then the cells die. So in this case, their hypothesis is that your housekeeping gene in your sample, even though if you treat the cell with drug, maybe you study patient with disease, you study animal with different phenotype, but the essential gene, the housekeeping genes, are so essential that your treatment shouldn't disrupt them. Because if you disrupt them, you probably have a dead sample. So in this case, they're going to look at the variation of this housekeeping gene across your sample. Because this gene shouldn't change, if you have a good measurement, this gene should be equal across all samples. But the fact that they might not be equal, they might be high and low. So this means that your different sample might have some bias. Some might be high, some might be low. just because of the technical problem. So they're going to use the stability or the heterogeneity of the expression level of housekeeping gene also as a normalization factor. And in this case, they're going to use a geometric mean. So now we see that they can apply two different concepts of average to different types of data. But what I want to show you is that these two are actually the same under the right circumstance. So just to recap, the average that we are used to is you sum everything together and then you divide it by the number of samples. The average that some of you might not be used to is the geometric mean, where you multiply all the data and then you take the nth root to average it out. So you can imagine that arithmetic mean is you sum and you divide to cancel out. For geometric mean, you multiply, so you have the root to average it out of the multiplication. So let's see how they are different in practice. Let's say you have two data points, 1 and 10,000, very far apart. Arithmetic mean is going to give you right in the middle, 5,000. Geometric mean, you multiply these two together and take the square root, and you're going to get 100. So it's going to be leaning toward the smaller value. Let's say you have 3 data points, 1, 10, and 100. Arithmetic mean, you're going to get about 37. For geometric mean, it's going to be 10. You multiply these, you get 1,000. You take the cube root, you get back 10. Again, geometric mean is leaning toward the smaller value. Why? So let's look at the situation that makes these two similar. We already saw a log normal distribution, right? The data itself is skewed, but once we log transform it, we get a normal distribution. So the good thing about logarithm is if you have a sum of logarithm, it's the logarithm of the product. Hopefully you remember this. And if you divide the logarithm, it's the logarithm of the nth root. So basically you have this identity that transforms the arithmetic mean of the log value of your data. So let's say your data are x, right? You take the log transform, you get a normal distribution, like the log x are a normal distribution. So you identify the mean by summing them and take the average. This is equivalent to if you don't transform your data, you don't touch your data, but you multiply them together and take the nth root, and you take the log, you get back the same thing. So basically geometric mean and arithmetic mean are equivalent, but they operate on the same data. And arithmetic mean operate on the log transform data, which are normally distributed, but geometric mean operate on the original data, which are still skewed. So now that we know this, let's go back and see what happens there. So for the negative control, because these are not signal, these are noise, non-specific binding, they are assuming that this is not going to be log normal, because these are not real intensity, not molecule count. These are background. So they assume that the background, you don't have to transform them. You assume that they're normally distributed, and you can use the arithmetic mean. But for these data, for the positive control and for the housekeeping, these are actual read count. So these read count, you can see the scale being very high, from 50 to 25,000. So these are skewed. So in this case, you have two choices. You can log transform them and you calculate the arithmetic mean. Or you don't transform them and you calculate the geometric mean. So in this case, the software just don't transform them and calculate the geometric mean. So they kind of make it confusing. But now you know why it's arithmetic mean versus geometric mean. Okay, so for the last section of today, we're going to talk about RNA sequencing. And this is probably what you are most excited for. It's something that has the highest probability that you're going to encounter in your work. Okay. So before we go into the detail, just want to remind that we have more than just coding RNA. We have non-coding RNA. And within coding RNA, there are also many species of RNA. So when we do RNA sequencing, in the past, all people thought of were messenger RNA. But nowadays, when we talk about RNA sequencing, people also wonder, do we have non-coding RNA in our sample? Like what kind of RNA are we analyzing? Can we identify these kind of non-coding but regulatory RNA that can explain our data? So what many people are doing today is called total RNA sequencing. Total in the sense that usually we just focus on messenger RNA. But now we want to sequence every RNA except ribosomal RNA. Ribosomal RNA is still the thing that no one wants. Because it's highly conserved, ribosome is highly conserved. So even if you sequence them, it's not going to tell you anything about biology most of the time. And even worse, in your RNA extract, 80% is going to be ribosomal RNA. So if you put in like 10,000 baht to sequence, you're going to have to lose 800 baht. If you put in 10,000, you need to lose 8,000 on sequencing ribosomal RNA. That doesn't say anything. So people have been developed several techniques to deplete or to remove ribosomal RNA. So because ribosomal RNA are very conserved, we know exactly what they are. So one thing you can do is people can design the probes against ribosomal RNA because we know the sequence. And we can use enzyme like RNA-seq that specifically target double-stranded RNA. So the only RNA in your sample that's going to be double-stranded will be ribosomal RNA because you put in probes for ribosomal RNA. So they're going to hybridize to ribosomal RNA and the enzyme is going to digest them. So in the end, hopefully, you're going to end up with everything but ribosomal RNA. And this will be called total RNA sequencing. But in certain situations, you want to focus on just messenger RNA, or you want to focus on certain group of RNA, like microarray. You can enrich for this. For messenger RNA, because RNA has the poly A tail, you can design a poly-T probes to pull them down. Very simple. For microRNA, because they are very small, like 50 to 25 nucleotide, I think, you can run the DNA sample and then select by the size. You can select the band that is the smallest size, and those will be your microRNA. So you can enrich for certain RNA as well. Or if you don't have any specific target, you can study the whole RNA species. So we mentioned a little bit before that when we want to study different RNA isoforms, so each gene can express multiple isoforms. And isoform happens when we have a splicing from one exon to the other exon that might not be next to each other. Sometimes you don't care about this. You just want the gene expression. So whether you detect isoform A, B, or C doesn't matter. But sometimes certain disease or certain regulatory mechanisms operate on the isoform. You get a switch from isoform 1 to isoform 2, and you get different function or different tissue type, for example. If you want to study this, then we want our sequencing strategy to be able to map this junction where one exon ends and the next exon begins. We already mentioned that with short read, this is going to be very challenging because the chance that your read are going to map to this location is very small. With pair-end sequencing, you get a better chance. Your forward read might be in exon 1 and your reverse might be in exon 2. Then you know that there is a splicing between exon 1 and exon 2, even though you don't have the read mapping exactly at the junction. But if you use long read sequencing, then you get the whole molecule. So just to show you, this is from 10 years ago, where people still deciding what kind of a sequencing strategy is good for isoform detection. So in this analysis, they try changing the read length, the length of the read, from 25 to 100. And they also try to do single end versus pair end. So these are two samples, one single end, one pair end, and another sample single end and pair end. What you can tell at a glance is when you switch from single end to pair end, with single end you have to have the read map to the junction. With pair end, as long as it's cover the junction, you are good. So by switching from pair end, single end sequencing to pair end, you get twice as many isoforms detected. So that's a good sign. And nowadays we are doing pair end sequencing. And as you increase your sequencing length, you're going to be able to map the location of your read kind of more specifically. So you also gain more isoform as well. You can detect more and more isoforms as you sequence longer and you do pair sequencing. But nowadays, now that we have long read data, nothing beats getting a full-length transcript sequence from long read information. So it depends on how much you want to be able to distinguish between isoforms. You can pick your technology accordingly. Okay, so what can we do with RNA-seq data? There are three ways that people proceed for RNA sequencing data. If you have a reference genome, you know the genome, you know the gene location, you can map your RNA sequencing on the genome. If you have a reference transcriptome, so reference transcriptome means that other people who have done RNA sequencing before have mapped out all the genes, all the isoforms. They can create a database of isoforms. So it's not DNA, it's not no intron. So the difference between genome reference and the reference transcriptome is in the reference genome, you have the exon and intron. So your analysis still have to be able to map out the exon and the intron. But if you use transcriptome as a reference, everything has no intron. So you are mapping your RNA sequencing data directly to the isoform. And in the third case, which might be rare for you, if you are studying non-model organisms, you have no reference genome, you have no reference transcriptome, so you're going to have to do some assembly. You're going to take your RNA sequencing read. So imagine it's the same as assembly for the genome. When you study a new species for the first time, you sequence the DNA, you have to put the DNA together in a graph to try to figure out what the chromosomes are. In the case of RNA sequencing, if we have no information at all, we also want to put the RNA read together, to assemble them together. But the final target is no longer a chromosome, it's going to be just one isoform. So hopefully we can map all the reads, kind of connect them together, so that we reconstruct the whole gene isoform. And when there are multiple splicing possibilities, then that means we have multiple parts of the graph. So this might be isoform 1, and this might be isoform 2. It's the same concept as DNA assembly. So there's nothing new here. We have to do. What we want to focus on, which is the choice that you have when you are studying model organisms. You have a choice of using the reference genome or reference transcriptome. Like in human, in mouse, you're going to have the choice. What's the difference? Let's say we look at this diagram first. In the genome, you have exon, intron, and exon. In the transcriptome, when the splicing is already performed, you lose the intron. So the molecules that you are sequencing, the transcript, have no intron. Most of them have no intron. This is your read. So your read is going to come from a certain part of your transcript. So this is the reference genome. This is the transcript molecules, the RNA molecules. This is your read. So if you are considering that you have two possibilities, mapping your read to the genome or mapping your read to the transcriptome, which one is easier? Of course, mapping to the transcriptome. Because your read and the transcriptome, that both of them do not have intron. So the alignment is going to be ungapped alignment. You assume that your read is going to be a certain region on the transcript. But if you want to align your read to the genome, we have seen this before. The software is going to have to assume that if the read map to a splicing junction, the first part of the read might map to the end of one exon, but the latter half of the read might map to the beginning of another exon. So the algorithm has to consider this possibility. So you can imagine that aligning to the genome is going to be slower because there is this possibility that it has to consider all the time. But if you are aligning to the transcriptome, it's going to be very fast. And we'll see how fast it is next lecture. It's fast enough that you turn from 8 hours to 30 minutes. That's fast. So just to give you some names of the tools that we will encounter in some assignment and maybe in your future. If you align your RNA sequencing data against the transcriptome very fast, there are specialized tools for that called Salmon and Callisto. And we'll talk about what they do next lecture. But if you align to the genome, so this is a classical approach, there is at least two software that I recommend, STAR and HiSat2. So these are going to have to consider the fact that your read might be separated into two portions, each mapped to a different exon. Okay. So when let's say we choose to align to the genome, so what we are going to need in order to help the alignment, because if we want to consider the fact that our reads, the reads here, come from protein coding gene, and some of them might span the splice junction. If our reference genome, if we don't have this location for the exon, it's going to be very difficult for the software to know, does this alignment make sense? Like, do we know that it's the end of one exon? If we don't know that there are exons there. So one thing that these tools will ask from you is the annotation. On your genome, where are the exons? Where are the genes? So these files will come in what's called a GTF or GFF format. So these are again just table files. In there will be chromosomes, in this case chromosome X, and then this will be the information whether it's gene or transcript or exon to tell you kind of which genomic elements are at that location. And then the base pair from this base pair to this base pair. And is it on the plus strand or the minus strand? And if it's a gene or exon, they're going to also annotate the gene ID for this location. So these are files that's already pre-made for all model organisms. You can download them from NCBI or from Ensemble. So when you run the sequence RNA-seq, when you align it to the genome, the software is going to ask you for the reference genome, the DNA sequence, and also for the annotation, so that the software knows where the exons are, whether you read map to exon or not exon. That makes a difference for RNA sequencing. Okay. So just to recap, this is what we covered today. We talked about three platforms: microarray, Nanostring, and RNA sequencing. The key trade-off is, so RNA sequencing is the most expensive, but you can detect any RNA molecules. You have a chance to discover something new. With Nanostring and microarray, you are restricted to the choice of genes or exon that's already decided. But it's good if you don't need to discover new RNA. You want to use it on your patient, for example, and you already know all the genes. You want to use it on you want to make it cheap for your patient. So in this case, microarray would be the cheapest platform. But it doesn't provide very accurate quantification of the gene expression. So to get the most accurate measurement, Nanostring gives you that, but it has the lowest number of genes that can be targeted. So you can see it's a trade-off between discovery against targeted, and the accuracy of the measurement against price. And that's the choice that we have to make for both platforms. Okay, so to give you a preview of what we will cover for the next lecture, we're going to go back to the RNA sequence alignment, not with the reference genome, but with the reference transcriptome. And we will talk about why it's so fast and what we gain from that speed. It's not just the time that we gain. We're going to gain something more beneficial. We're going to talk about once we have the data, what are the units for gene expression. And different units will have different use, different utilization. We're going to talk in more details about the statistical model for gene expression data from RNA sequencing. We already saw the log normal distribution model for microarray data and the Nanostring data. But for RNA sequencing, the read count, these are integers, so they are not really normal. And because the read count are not as high as the number of molecules, they also cannot be approximate as log normal either. So we have to settle for a new model. I'm not sure if you have seen this before, but next class we're going to talk about the negative binomial distribution and how people use it to capture gene expression data for the read count from RNA sequencing. And then we'll talk about the statistical model that we use to identify differentially expressed genes, which genes are upregulated or downregulated in your sample. Okay, so that will be the next time. Okay,